Because the calcium level is low, a small change in absolute numbers of ions represents a relatively large percentage change in calcium concentration. In response to a signal relayed by a signal transduction pathway, the cytosolic calcium level may rise, usually by a mechanism that releases Ca2+ from the cell's ER. The pathway leading to calcium release involves still other second messengers, diacylglycerol (DAG) and inositol triphosphate (IP3). These two messengers are produced by cleavage of a certain kind of phospholipid in the plasma membrane. Figure shows how this occurs and how IP3 stimulates the release of calcium from ER. Because IP3 acts before calcium in the pathway, calcium could be considered a third messenger. However, scientists use the term second messenger for all small, non-protein components of signal transduction pathways. Calcium ions activate a signal transduction protein directly, but often they function by means of calmodulin, a Ca2+ binding protein present at high levels in eukaryotic cells. In an animal cell, for example, calmodulin may represent as much as 1% of the total protein. Calmodulin mediates many calcium-regulated processes in cells. When calcium ions bind to it, calmodulin changes conformation and then binds to other proteins, activating or inactivating them. The proteins most often regulated by calmodulin are protein kinases and phosphatases, the most common relay proteins in signaling pathways. 6. Cellular responses to signals. We now take a closer look at the cell's eventual response to an extracellular signal, what some researchers call the output response. What is the nature of the final step in a signaling pathway? A. In response to a signal, the cell may regulate activities in the cytoplasm or transcription in the nucleus. Ultimately, a signal transduction pathway leads to the regulation of one or more cellular activities. The regulated activities may occur in the cytoplasm, such as a rearrangement of the cytoskeleton, the opening or closing of an ion channel in the plasma membrane, or some aspect of cell metabolism. As we have discussed already, the response of liver cells to signaling by the hormone epinephrine helps regulate cellular energy metabolism. The final step in the signaling pathway activates the enzyme that catalyzes the breakdown of glycogen. Other signaling pathways ultimately regulate not the activity of an enzyme but the synthesis of enzymes or other proteins, usually by turning specific genes on or off. Genes in a cell's DNA function by being transcribed into an RNA version called messenger RNA, mRNA, which leaves the nucleus and is translated into a specific protein by ribosomes in the cytoplasm. Special protein called transcription factors control which genes are turned on, that is, transcribed into mRNA in a particular cell at a particular time. The activity of a transcription factor may itself be regulated by a signaling pathway that extends into the cell nucleus. All the kinds of signal receptors and relay molecules introduced in this chapter participate in gene-regulating pathways, as well as in pathway leading to other kinds of responses. The molecular messengers that produce gene regulation responses include growth factors and certain plant and animal hormones. B. Elaborate pathways amplify and specify the cell's response to signals. Why are there often so many steps between the original signaling event at the cell surface and the cell's response? As mentioned earlier, signaling pathways with a multiplicity of steps have two important benefits. They amplify the signal and thus response, and they contribute to the specificity of response. C. Signal amplification. Elaborate enzyme cascade amplifies the cell's response to signal. At each catalytic step in the cascade, the number of activated products is much greater than in the preceding step. For example, in the epinephrine-triggered pathway in figure, each adenylate cyclase molecule catalyzes the formation of many CAMP molecules, each molecule of protein kinase phosphorylate many molecule of next kinase in the pathway, and soon. The amplification effects depends the fact that these protein persists in active form long enough to process numerous molecules of the substrate before they become inactive again. As a signal amplification, a small number of epinephrine molecule binding to receptors on the surface of liver cell or skeletal muscle can lead to a release of hundreds of millions of glucose molecule from glycogen. D. The specificity of cell signaling. Consider two different cells in your body, a liver cell and heart muscle cell, for example. Both are in contact with your bloodstream and are therefore constantly exposed to many different hormone molecules, as well as to local regulators secreted by nearby cells. Yet the liver cell responds to some signals but ignores others, and the same is true for the heart cell. And some kinds of signals trigger responses in both cells but different responses. Epinephrine stimulates the liver cell to break down glycogen, but the main response of the heart cell to epinephrine is contraction, leading to a more rapid heartbeat. How do we account for this difference? Each of the specificity exhibited in cellular responses to signals is the same as the basic explanation for virtually all differences between cells. Different kinds of cells have different collections of proteins, figure 9. The response of a particular cell to a signal depends on its particular collection of signal receptor proteins, relay proteins, and proteins needed to carry out the response. A liver cell, for example, is poised to respond appropriately to epinephrine by having the proteins, as well as those needed to manufacture glycogen. Thus, two cells that respond differently to the same signal differ in one or more of the proteins that handle and respond to the signal. Notice in figure 9 that pathways may some molecules in common. For example, cells A, B, C all use the same receptor protein for the triangular signal molecule. Differences in other proteins account for their differing responses. That in cell B, a pathway triggered by a single kind of signal diverges to produce two responses, and that in cell C, two pathways triggered by separate signals converge to modulate a single response. Branching of pathways and crosstalk between pathways are important in regulating and coordinating a cell's responses to incoming information. Moreover, the use of some of the same proteins in more than one pathway allows the cell to recognize a number of different proteins it must make. The importance of relay proteins that serve as points of branching or intersection in signaling pathways is highlighted by the problems arising when these proteins are defective or missing. For instance, in an inherited disorder called Wiskott-Aldrich syndrome, the absence of a single relay protein leads to such diverse effects as abnormal bleeding, eczema, and a predisposition to infections and leukemia. These symptoms are thought to arise primarily from the absence of the protein in cells of the immune system. By studying normal cells, scientists found that the WIS protein is located just beneath the cell surface. The protein interacts both with microfilaments of the cytoskeleton and with several different components of signaling pathways that relay information from the cell surface, including pathways regulating immune cell proliferation. This multifunctional A protein is thus both a branch point and an important intersection point in a complex signal transduction network that controls immune cell behavior. When the WIS protein is absent, the cytoskeleton is not properly organized and signaling pathways are disrupted, leading to the WIS symptoms. To keep figure 9 simple, we have not indicated the inactivation mechanisms that are an essential aspect of cell signaling. For a cell of a multicellular organism to remain alert and capable of responding to incoming signals, each molecular change in its signaling pathways must last only a short time. As we saw in the cholera example, if a signaling pathway components become locked into one state, whether active or inactive, dire consequences for the organism can result. Thus a key to a cell's continuing receptiveness to regulation is the reversibility of the changes that signals produce. The binding of signal molecules to receptors is reversible, which means that the lower the concentration of signal molecules, the fewer will be bound at any given moment. When the receptor cell reverts to its inactive form, then, by a variety of means, the relay molecules return to their inactive forms. The GTPase activity intrinsic to AG protein hydrolyzes its bound GTP. The enzyme phosphodiesterase converts CAMP to AMP. Protein phosphatases inactivates phosphorylated kinases and other proteins, and so forth. As a result, the cell is soon ready to respond to a fresh signal. A3. Signal transduction in bacteria and bacterial chemotaxis. Prokaryotic signal transduction systems and a few eukaryotic pathways use phosphotransfer schemes involving two conserved components, a histidine protein kinase and a response regulator protein. Protein kinase, which is regulated by environmental stimuli, autophosphorylates at a histidine residue, creating a high-energy phosphoryl group that is subsequently transferred to an aspartate residue in the response regulator protein. Phosphorylation induces a conformational change in the regulatory domain that results in activation of an associated domain that affects the response. The basic scheme is highly adaptable, and numerous variations have provided operation within specific signaling systems. The domains of two-component proteins are modular and can be integrated into proteins and pathways in a variety of ways, but the core structures and activities are maintained. Thus, detailed analyses of a relatively small number of representative proteins provide a foundation for understanding this large family of signaling proteins. Chemotaxis. Chemoreceptor on bacteria detects attractant and repellent and sends signal to flagella. Some of these molecules as aspartate bind directly to chemoreceptors, whereas others are carried by binding proteins. The direction of rotation of flagella determines whether bacteria swim smooth or tumble. Bacterium flagella is an extracellular appendage that is rotated by a motor located at the junction of flagellum and cell envelope. It is powered by the proton motive force across the plasma membrane. An intriguing feature of this motor is that it can rotate clockwise or counterclockwise, and switch between them almost instantaneously. When the flagella rotate counterclockwise, helical filaments form a coherent bundle and cell swims smoothly. When the flagella rotate clockwise, in contrast, the bundle flies apart because the screw sense of the helical flagella does not match the direction of rotation. Each flagellum then pulls in a different direction, and so the bacterium tumbles. When a bacterium moves toward an increasing concentration of an attractant, tumbling becomes less frequent. In contrast, when it moves away from the attractant, tumbling becomes more frequent. Repellent have opposite effect on tumbling frequency. Tumbling serves as reorient a misdirected bacterium. Bacteria possess temporal sensing mechanism, i.e. a bacterium detects the spatial gradient of attractant not by comparing the concentration at its head and tail, but by traveling through space and comparing its observations through time. Chemotaxis begins with the binding of molecules to four kinds of receptors in the plasma membrane. E. coli and other gram-negative bacteria, the PM is surrounded by an outer membrane that is permeable to small molecules. A paraplasmic space separates the PM from the outer membranes. Receptors are encoded by the TSR, TAR, TRG and TAP genes and were originally called methyl-accepting chemotaxis proteins, MCPs, because they are reversibly methylated. Repellents and some attractants bind to paraplasmic proteins, which then interact with the receptors. For e.g., the attractant aspartate binds directly to TAR protein, which is also known as aspartate receptor. The four chemoreceptors have the same design. Each approximately 60 kb protein contains four regions. 1. A paraplasmic domain that binds an attractant or a repellent. 2. A transmembrane segment consisting of two alpha helices. 3. A cytosolic system, and 4. A transmembrane region that can be reversibly methylated at several sites. Their cytosolic domains are highly conserved because they interact with the same components of the processing system. In fact, the cytosolic domains of TSR and TAR contain an identical 48 residue sequence. In contrast, their paraplasmic domains are distinctive because they bind different attractants, repellents, and attractant protein complexes. A chimeric receptor formed from the amino terminal half of TAR and the carboxy terminal half of TSR is triggered by aspartate and other attractant that are normally sensed by TAR. Thus, these chemoreceptors transmit signals across the membrane in essentially same way. The dimensional structure of paraplasmic domain of aspartate receptor dimerizes to form a highly elongated structure. Each subunit is 4 helix bundle that is 20 AU in diameter and more than 70 AU long. The binding sites for aspartate are at the interface between subunits, more than 60 AU from the membrane surface. A reciprocal phosphorylation cascade controls the direction of flagellar movement. Genetic analysis revealed the existence of common processing genes and provided valuable clues concerning its molecular circuitry. H have been identified. A, B, C, D, W, R, Y, and Z mutation in these genes impair chemotaxis to all attractant and repellent. In contrast with mutation in a particular kind of receptor will block responses to a subset of these agents. Bacteria void of all cytosolic J proteins, known as gutted strains, swim smoothly all the time, because their flagella rotate counterclockwise CCW continuously. The addition of J proteins to gutted stains, by controlling expression of J gene, lead to increased clockwise CW rotation. Furthermore, flagella rotates exclusively CW in mutants that express abnormally high levels of J protein in the presence of normal amounts of the other J proteins. These finding indicates that the direction of flagella rotation is controlled by the JY protein. The essentiality of ATP for both the excitation and adaptation phases of chemotaxis provides a tantalizing hint as to how JY might be controlled. A specific aspat trait of JY can become phosphorylated. Most significant, phosphorylated J induces tumbling, whereas dephosphorylated J has virtually no effect on flagellar switch. Phosphorylated J is transient species. These attached phosphoryl group is simultaneously hydrolyzed in a reaction that is increased 100-fold by JZ. How does J acquire phosphate group and how its phosphorylation controlled by chemoreceptor? Melsiman found that purified G protein undergoes a slow autophosphorylation. The site of phosphorylation is N3 of histidine residue. This phosphorylation is greatly accelerated by the binding of G, together with JW, to an unoccupied receptor or a repellent receptor complex but not to an attractant receptor complex. The phosphoryl group attached to G can then be transferred to J. Hence, repellent increase the level of phosphorylated G and, in turn, of phosphorylated J. Attractant have the opposite effect. In this way, repellent increase CW rotation and tumbling, whereas attractants increase CCW rotation and smooth swimming. It is interesting to note that G belongs to a family of regulatory protein that enables the bacteria to sense their environment and internal metabolic state. For e.g., enter B, which is a homolog of G, controls the expression of genes that respond to nitrogen balance in cell. All homologs of G are autophosphorylating kinases that regulate the activity of target proteins by phosphorylating them. These sensor effector pairs are called two-component regulatory systems. Quorum sensing in bacteria and other organisms. Quorum sensing is a type of decision-making process used by decentralized groups to coordinate behavior. Many species of bacteria use quorum sensing to coordinate their gene expression according to the local density of their population. Similarly, some social insects use quorum sensing to make collective decisions about where to nest. In addition to its function in biological systems, quorum sensing has several useful applications for computing and robotics. Function is a decision-making process in any decentralized system, as long as individual components have a means of assessing the number of other components they interact with, and b a standard response once a threshold number of components is detected. Bacteria. So the best-known examples of quorum sensing come from studies of bacteria. Bacteria use quorum sensing to coordinate certain behaviors based on the local density of the bacterial population. Quorum sensing can occur within a single bacterial species as well as between diverse species, and can regulate a host of different processes, essentially serving as a simple communication network. A variety of different molecules can be used as signals. Mechanism. Bacteria that use quorum sensing constantly produce and secrete certain signaling molecules called autoinducers or pheromones. These bacteria also have a receptor that can specifically detect the signaling molecule inducer. When the inducer binds the receptor, it activates transcription of certain genes, including those for inducer synthesis. There is a low likelihood of a bacterium detecting its own secreted acetylhomoserine lactone al. Thus, in order for transcription to be activated, the cell must encounter signaling molecules secreted by other cells in its environment. When only a few other bacteria of the same kind are in the vicinity, diffusion reduces the concentration of the inducer in the surrounding medium to almost zero, so the bacteria produce little inducer. However, as the population grows, the concentration of the inducer passes the threshold, causing more inducer to be synthesized. This forms a positive feedback loop, and the receptor becomes fully activated. Activation of the receptor induces the upregulation of other specific genes, causing all of the cells to begin transcription at approximately the same time. This coordinated behavior of bacterial cells can be useful in a variety of situations. For instance, the bioluminescent luciferase produced by B. fischeri would not be visible if it were produced by a single cell. By using quorum sensing to limit the production of luciferase to situations when cell populations are large, B. fischeri cells are able to avoid wasting energy on the production of useless products. Examples. Fibrifera. Quorum sensing was first observed in Vibrio fischeri, a bioluminescent bacterium that lives as a mutualistic symbiont in the photophore or light-producing organ of the Hawaiian bobtail squid. When B. fischeri cells are free-living or planktonic, the autoinducer is at low concentration and thus cells do not luminesce. However, when they are highly concentrated in the photophore, about 10-11 cells per milliliter, transcription of luciferase is induced, leading to bioluminescence. Escherichia coli. In the gram-negative bacteria Escherichia coli, cell division may be partially regulated by autoinducer AI2-mediated quorum sensing. This species uses AI2, which is produced and processed by the LSR operon. Part of it encodes an ABC transporter which imports AI2 into the cells during the early stationary, latent phase of growth. AI2 is correlated by the LSRK kinase, and the newly produced phospho-AI2 can either be internalized or used to suppress LSRR, a repressor of the LSR operon, thereby activating the operon. Transcription of the LSR operon is also thought to be inhibited by dihydroxyacetone phosphate, DHAP, through its competitive binding to LSRR. Glyceraldehyde 3-phosphate has also been shown to inhibit the LSR operon through CAMP-CAPK-mediated inhibition. This explains why when grown with glucose E. coli will lose the ability to internalize AI2 because of catabolite repression. When grown normally, AI2 presence is transient. Pseudomonas aeruginosa. The opportunistic bacteria Pseudomonas aeruginosa uses quorum sensing to coordinate the formation of biofilms, swarming motility, exopolysaccharide production, and cell aggregation. These bacteria can grow within a host without harming it, until they reach a certain concentration. When they become aggressive, their number is sufficient to overcome the host's immune system and form a biofilm, leading to disease. In this, AI2 was found to increase expression of SDIA, a transcriptional regulator of promoters which promote FTSQ, part of the FTSQ-AZ operon essential for cell division. Another form of gene regulation which allows the bacteria to rapidly adapt to surrounding changes is through environmental signaling. Recent studies have discovered that anaerobiosis can significantly impact the major regulatory circuit of QS. This important link between QS and anaerobiosis has a significant impact on production of virulence factors of this organism. It is hoped that the therapeutic enzymatic degradation of the signaling molecules will prevent the formation of such biofilms and possibly weaken established biofilms. Disrupting the signaling process in this way is called quorum quenching. Social insects. Social colonies are an excellent example of a decentralized system, because no individual is in charge of directing or making decisions for the colony. Several groups of social insects have been shown to use quorum sensing when making collective decisions. Examples. Ants. Colonies of the ant Temnothorax albipinus nest in small crevices between rocks. When the rocks shift and the nest is broken open, these ants must quickly choose a new nest to move into. During the first phase of the decision-making process, a small portion of the workers leave the destroyed nest and search for new crevices. When one ant finds a potential nest, she assesses the quality of the crevice based on a variety of factors including the size of the interior, the number of openings based on light level, and the presence or absence of dead ants. The worker then returns to the destroyed nest, where she will wait for a short period before recruiting other workers to follow her to the nest she found using a process called tandem running. The waiting period is inversely related to the quality of the site. For instance, a worker that has found a poor site will wait longer than a worker that encountered a good site. As the new recruits visit the potential nest site and make their own assessment of its quality, the number of ants visiting the crevice increases. During this stage, ants may be visiting many different potential nests. However, because of the differences in the waiting period, the number of ants in the best nest will tend to increase at the greatest rate. Eventually, ants in this nest will sense that the rate at which they encounter other ants has exceeded a particular threshold, indicating that the quorum number has been reached. Once the ants sense a quorum, they return to the destroyed nest and begin rapidly carrying the brood, queen, and fellow workers to the new nest. Scouts tandem running to other potential sites are also recruited to the new nest and the entire colony moves. Thus, although no single worker may have visited and compared all of the available options, quorum sensing enables the colony as a whole to quickly make good decisions about where to move. Honeybees. Honeybees Apis mellifera also use quorum sensing to make decisions about new nest sites. Large colonies reproduce through a process called budding, in which the queen leaves the hive with a portion of the workers to form a new nest elsewhere. After leaving the nest, the workers form a swarm that hangs from a branch or overhanging structure. This swarm persists during the decision-making phase until a new nest site is chosen. The quorum sensing process in honeybees is similar to the method used by Temnothorax ants in several ways. A small portion of the workers leave the swarm to search out new nest sites, and each worker assesses the quality of the cavity she finds. The worker then returns to the swarm and recruits other workers to her cavity using the honeybee waggle dance. However, instead of using a time delay, the number of dance repetitions the worker performs is dependent on the quality of the site. Workers that found poor nests stop dancing sooner, and can therefore be recruited to the better sites. When visitors to a new site sense that a quorum number, usually 10 to 20 bees, has been reached, they return to the swarm and begin using a new recruitment method called piping. This vibration signal causes the swarm to take off and fly to the new nest location. In an experimental test, this decision-making process enabled honeybee swarms to choose the best nest site in 4 out of 5 trials. Plant two-component signaling system. The prokaryotic lifestyle of plants. At first glance, a 100-year-old tree and a bacterium do not have much in common. However, both plants and bacteria face a similar problem. They are confined to their environment and have to endure and adapt to a broad array of environmental conditions. To manage this complex, prokaryotic organisms rely on a two-component signaling system, which is involved in such diverse processes as chemotaxis, osmotic sensing, and light perception. Stock et al. 2000. As shown in recent years, all canonical elements of two-component signaling systems are also found in plants. Schaller et al. 2001. This update focuses on recent advances in our understanding of plant two-component systems, with particular emphasis in the function of plant response regulators. Protein elements and biochemistry of two-component signaling systems. In its simplest form, the two-component signaling system is composed of 2 elements, a His kinase and a response regulator. Figure 1a. Although the prokaryotic two-component signaling systems are composed of just these 2 proteins, there also exist more complex signaling systems that include additional elements, which are referred to as multi-step two-component systems. Figure 1b. Chemistry two-component signal transduction is a phosphorylate. After a signal-induced activation by the input domain, the transmitter of a His kinase autophosphorylates at a conserved His residue. Figure 1a. The phosphorylate group is then transferred to an invariant ASP within a conserved domain of the receiver. Figure 1a. This phosphorylate controls the activity of the output domain of the response regulator, which results, for example, in interaction with other proteins or binding to DNA. Figure 1. Schematic representation and basic features of the symbol A and the multi-step B two-component signaling systems. Signal perception by the input domain of the His or hybrid kinase induces autophosphorylation of the transmitter at a conserved His residue H. The 5P is relayed to a conserved ASP residue D that is localized either in the receiver of the cognate response regulator A or, in the case of hybrid kinases, in an attached receiver domain B. In the multi-two-component signaling system, His containing phosphotransfer HPT domain proteins serve as phospho His intermediates between the hybrid kinase and the response regulator. Aligned one-step, two-component signaling system can be extended into a multi-step system by including additional protein modules containing phosphorylatable ASP or His residues. The second ASP is again included in a receiver module. In plants, the additional receiver module is often carboxydermally fused to the His kinase, thereby creating a hybrid type of sensor kinase. Figure 1b. The second is embedded in a domain known as the HPT domain. The HPT domain enables the transfer of a phosphoryl group between 2 receiver modules. The prokaryotic HPT domains are physically linked to other two-component signaling elements, but the Arabidopsis HPT domains are all individual proteins. Many of the prokaryotic HPT domains are physically linked to other two-component signaling elements, but the Arabidopsis HPT domains are all individual proteins. Why are some two-component systems of the simple and others of the complex type? The reason for extending the complexity of two-component systems does not apparently lie in signal amplification. Within a two-component system, the signal is relayed as a phosphoryl group in a strict one-to-one manner. Thus, the signaling output of the simple and the multi-step two-component system is the same. If it is not the gain in signal amplification, what could be the reason behind the apparent preference of multi-step systems in plants? The answer to this question will become clear in subsequent sections. Two-component signaling elements of Arabidopsis. In the early 1990s, the cloning of 3 genes led to the breakdown of a dogma. Until then, it was believed that two-component signaling systems were restricted to prokaryotes. With the realization that the Arabidopsis ethylene receptor ETR1, the phytochrome photoreceptors, and the yeast Saccharomyces cerevisiae, Osman sensor SLN1 were all related to His kinases, it became clear that some genera of eukaryotes depended on two-component systems as well. Arabidopsis genome now completed. 17 His and hybrid kinase-like proteins AHK, 5 HPT proteins AHP, and 23 response regulators ARR have been identified in Arabidopsis. Although analysis has begun, it is evident that, with the exception of phytochromes, most AHKs, AHPs, and ARRs in principle function as phosphotransfer proteins. Ethylene perception and signaling. The ethylene perception and transduction system is one of the best understood plant signaling pathways. The indication of ETR1, figure 2a, was followed by the description of 4 additional ethylene receptors either belonging to the hybrid kinase type, ETR2 and IN4, or the His kinase type, HERS1 and HERS2. Ethylene binding sites in a heterologous yeast system and in genetic studies, it was elegantly shown that ETR1 is a membrane-associated ethylene receptor, figure 2a. ETR1 exhibits His autophosphorylation activity in vitro and interacts with several AHP proteins in the yeast-2 hybrid system. However, it has not been demonstrated that ETR1 and the other members of the ethylene receptor family act as ethylene-triggered hybrid kinases in plants. Figure 2. Picture of representative members of two-component signaling elements from Arabidopsis. A. Hybrid kinases, CRE1 and ETR1. The function of the GAF-related domain of ETR1 is unknown. ARR, ARR2, type B response regulator, ARR4, type A response regulator. The GARP motif of type B response regulators is responsible for DNA binding in the nuclear localization sequences, NLS, for nuclear targeting. The pro-NGL-enriched PQ-rich domain serves as transactivation domain, which may interact with the basal transcription machinery. C, AHP, AHP1 and AHP2. The conserved amino acid residues H, His, T, ASP of the different two-component elements are indicated. Ethylene signaling through a two-component system, one would expect to identify mutants affected in genes coding for HPT domain proteins and response regulators. Such mutants have not been reported, potentially because of functional redundancy within these signaling elements. The element shown downstream of the ethylene receptors and found capable of physically interacting with ETR1 and HERS1 is SENDER1, constitutive triple response 1, which is thought to function as RAS-like mitogen-activated protein kinase. Thus, it appears that the hybrid kinase-like ethylene receptors can function in a unique way, linking the perception of the hormone to the activity of a mitogen-activated protein kinase cascade. However, although a central component in ethylene signal transduction, there are several indications for the existence of additional signaling elements. Sender-1 null mutants are still slightly responsive to ethylene, and B, the phenotype of quadruple ETR1 ETR2 IN4 ERS2 receptor mutants is more pronounced in comparison with that of Sender-1. With AHP and ARR knockout lines now in hand, it should soon be possible to determine whether two-component proteins represent these additional ethylene signaling components. Recent advances in our understanding of cytokinin signaling offer important insights into plant two-component systems. In 1996, a putative hybrid kinase, CKI1, was described by Kakamoto, which, when overexpressed, resulted in cytokinin-independent growth of Kali. However, most of the important advances came just recently. By for the loss of cytokinin-independent greening and shoot formation of hypocodal X plants from Arabidopsis, the CRE1 cytokinin response 1 mutant was identified. The CRE1 gene codes for a hybrid kinase with an immunoterminal cytokinin binding domain flanked by two transmembrane stretches, figure 2a. In contrast to ETR1, CRE1 contains a second receiver-like domain downstream of the transmitter, figure 2a. When it's fission yeast, Schizosaccharomyces convat strains, in which the endogenous hybrid kinases were knocked out, the CRE1 gene product rescued the altered growth phenotypes of these mutants in the presence of cytokinin. These results indicate that CRE1 can bind cytokinin, B, cytokinin binding induces the His kinase activity of CRE1, and C, CRE1 can transfer the phosphoryl group to the intrinsic HPT domain proteins of yeast and fission yeast initiating multi-step, two-component signaling cascades. Function of CRE1 as a sensor His kinase in plant is also suggested by recent studies performed in Arabidopsis protoplasts. However, although CRE1 is a cytokinin receptor with His kinase activity, it is probably not responsible for mediating all of the plant's cytokinin responses. Two additional CRE1-like His kinases in Arabidopsis, AHK2 and AHK3, presumably also function as cytokinin receptors. Arabidopsis response regulators and HPT domain proteins. Although like proteins were described in the early 1990s, it required almost a decade for other two-component signaling elements to be identified in higher plants, namely response regulators and HPT domain proteins. For sequence homology, domain composition, and transcriptional regulation, most Arabidopsis ARRs can be described as either type A or B. The type A response regulators are relatively small and contain a receiver domain along with short C-terminal extensions, figure 2b. The mRNA of type A ARRSI is rapidly accumulating to varying extent after cytokinin treatment. The type B ARRs have receiver domains along with an extended C-terminal output domain and are not induced by cytokinin, figure 2b. Receptor array studies, however, indicate that the expression of the type B response regulator ARR2 is downregulated by the phytochroma photoreceptor in seedlings. We will now highlight recent insights into the function of type A and type B ARRs. Type B ARRs, response regulators with transcription factor properties. Comparison of the amino acid sequences of the output domains of type B ARRs revealed a highly conserved stretch of about 60 amino acids. The stretch was given the name GARP motif according to the founding members Golden 2, arranged by, and PSR1 figure 2b. GARP motif is predicted to fold into an helical structure and is related to the DNA binding motifs of the basic helix, loop helix, and the MYB classes of transcription factors. Thus, it was proposed that the GARP motif represented a novel DNA binding domain, and that type B ARRs might function as transcription factors. By studying the subcellular distribution and transactivation capability in vivo, it became evident that type B ARRs localized to the nucleus and were transcriptionally active. The first pointing to potential target promoters came with the histochemical analysis of the expression pattern of an ARR2 promoter, UIDA construct in transgenic Arabidopsis plants. Although the ARR2 gene was expressed in almost all Arabidopsis organs, particularly high glucuronidase activity was observed in pollen grains. A similar expression pattern is described for the nuclear genes for components of the mitochondrial respiratory complex I, NCI genes. The pollen-specific activity of the NCI genes is mediated by a conserved promoter element called the pollen box. Lorman and colleagues demonstrated sequence-specific binding of ARR2 to the pollen box element and narrowed down the binding sequence to a 5' W GAT W3 core, where W is either or T. The sequence matches the consensus binding site identified independently by Sakai et al. 2000 using the GARP domains of ARR1 and ARR2 in an oligoselection approach. With the addition of the AI-genesis targets and with the demonstration of transactivation capability, it is evident that ARR1, ARR2, and presumably the other type B ARRs function as transcription factors inside the nucleus, figure 3. Figure 3. MAV and Arabidopsis multi-step phosphorylate, demonstrating signal transduction from the extranuclear membranes to the nucleus. Signal perception by the input domain of a membrane-bound sensor His kinase G. CRE1 induces autophosphorylation of the transmitter. The phosphate P is relayed to the cognate AHP, e.g. AHP2 interacting with the cytosolic receiver module of the kinase. The phosphorylated AHP shuttles to the nucleus and transfers the phosphate to the receiver domain of its cognate type B response regulator, e.g. ARR1 and ARR2. Dephosphorylated AHP shuttles back to the cytosol where it can be rephosphorylated. Phosphorylation of the receiver domain of the response regulator results in a conformational change in its output domain. This conformational change may either induce binding of type B ARRs to their target promoter sequence, e.g. the gap box in the case of ARR1 and ARR2, and/or activation of their transcriptional capacity. The promoter-associated, active response regulators triggers the transcription of its target genes, e.g. NCI genes, type A ARR genes. The conserved amino acid residues H, His, T, S of the different two-component modules are indicated. POLII, RNA polymerase II-dependent basal transcription machinery. Recent evidence suggests that the type B response regulators ARR1 and ARR2 are involved in cytokinin signaling. ARR1 null mutant was found to be partially resistant to cytokinin in shoot regeneration and root elongation, whereas ARR1 overexpressing lines displayed the opposite, hypersensitive phenotype. Symptoms and sensitivity to cytokinin were observed when the expression of several type A ARR genes including ARR4 were analyzed in the ARR1 mutant and ARR1 overexpressing lines. A cytokinin-inducible effect of ARR1 and ARR2 on the expression of an ARR6 type A ARR reporter construct was observed in Arabidopsis mesophyll protoplasts. These results suggest that ARR1 and ARR2 mediate the cytokinin-induced expression of type A ARRs, figure 4. It is of interest to note that ARR2 very strongly induced the ARR6 reporter gene expression in Arabidopsis protoplasts even in the absence of cytokinin. This observation is indicative for the presence of a further stimulus that enables the activation of ARR2 and, in turn, ARR2 regulated target genes in protoplasts. Figure 4. Model for the action of an ARR4 comprising two-component signaling system on phytochrome B signal transduction. Red light R induces the photoconversion of the inactive PR form of phytochrome B (PHYB) in the physiologically active PFR form. Phosphorylated ARR4 then stabilizes the active PFR form by inhibition of PHYB dark reversion dark reverend. PFR stabilization enhances the sensitivity of plants to red light and induces more intense photomorphogenic responses PHYB signaling. Phosphorylation state in the activity of ARR4 is potentially regulated by a multi-step phosphorylate comprising of the HPT protein AHP1 and presumably CRE1-like hybrid kinases two-component signaling. Therefore, ARR4 enables crosstalk and thus integration of signal information provided by hormone cytokinin and the red light signaling cascades crosstalk. The expression of ARR4 and the protein composition of the two-component system are regulated by cytokinin transcriptional regulation. The conserved amino acid residues H, His, T, S of the different two-component modules are indicated. FR, far red light, P, phosphate residue. From the Arabidopsis protoplasts, it is not clear whether the activities of ARR1 and ARR2 are regulated by AS phosphorylation. This raises the question of whether cytokinin signals through a canonical phosphorylate to regulate the transcription factor properties of type B response regulators. However, the conservation of the canonical phosphorylation sites in the type B ARRs and also the type A ARRs are strongly indicative of the functional relevance of these sites. Moore and colleagues 2001 provided in Planta evidence that the phosphoaccepting receiver domain suppresses the function of ARR1 in the absence of cytokinin and that the cytokinin signal releases this suppression. This release could potentially be induced by phosphorylation of the highly conserved ASP in the receiver domain of ARR1. The finding that type B ARRs are localized to the nucleus raises the question as to how the signal information perceived by the extranuclear and membrane-bound sensor His kinases is transduced to the nuclear compartment. For instance, the cognate sensor His kinases of ARR1 and ARR2 is very likely the cytokinin receptor CRE1, which is predicted to be present on an extranuclear membrane. Moreover, protein-protein interaction studies revealed that the sensor His kinases CKI1, CRE1, and ETR1 cannot directly interact with ARR1 and ARR2. As outlined in Figure 1b, the Arabidopsis HPT domain proteins could perform the information transfer as part of a multi-step, two-component phosphorylate. ETR1, CKI1, and CRE1, as well as response regulators ARR2, can interact with AHPs, indicating that HPT domain proteins may serve as molecular adapters. AHPs are localized to the cytosol as well as the nucleus of the plant cell, and thus have the ability to perform functions in both compartments. AHP proteins have a size of around 12 kD and may enter the nucleus by diffusion. At least some AHPs transiently accumulate in the nucleus of Arabidopsis protoplasts in response to cytokinin application. These observations support the following model of AHP action, Figure 3. After activation by the specific signal, the sensor has kinase autophosphorylates and then transfers the phosphoryl group to the cognate AHP. The AHP then shuttles into the nucleus and relays the phosphate to nuclear type B ARRs. Phosphorylation of the receiver module triggers the transcription factor properties of type B ARRs, which results in an altered activity of their target genes. The dephosphorylated AHP then moves back to the cytoplasmic compartment to be rephosphorylated. This indicate that higher plants have adapted the ancestral two-component systems to a novel type of eukaryotic intracellular signaling mechanism. Because necessity for a shuttling HPT domain protein, this adaptation could only be achieved by use of multi-step, two-component systems. Type A ARRs, response regulators modulating other signal transduction pathways. Mentioned above, the transcription of type A response regulator genes like ARR4 is rapidly induced by cytokinin. This and some additional criteria establish the hypothesis that type A ARRs are primary response genes of cytokinin. Since studies suggest that the cytokinin-regulated transcriptional control of type A ARR genes is mediated by type B response regulators, Figure 4. The protein recently was identified for a type A response regulator, thereby lending insight into the signaling mechanisms that operate downstream of these response regulators. Swear and colleagues 2001 demonstrated that ARR4 specifically interacts with the plant photoreceptor phytochrome B. It is interesting that ARR4 did not interact with the COOH terminus of phytochrome B, which is related to transmitter domains of His kinases, but with the extreme NH2 terminus. Phytochrome B is a red, far red reversible chromoprotein, which regulates red light-specific photomorphogenetic responses in plants. It is synthesized in the inactive PR form and photoconverted by red light into the physiologically active PFR form. Far red light is able to induce a photoconversion of the active PFR form back to the inactive PR form. In addition, active PFR converts back to inactive PR in a light-independent manner known as dark reversion, which is thought to be an additional switch-off mechanism for phytochromes. Which is in yeast and Arabidopsis indicated that the interaction of ARR4 with phytochrome B resulted in the stabilization of the physiologically active PFR form by inhibition of PFR to PR dark reversion, Figure 4. Functional relevance of the phytochrome B, ARR4 interaction and PFR stabilization was examined in transgenic plants overexpressing ARR4. These plants displayed a hypersensitivity to red light in respect to all photomorphogenic responses tested, e.g. hypocodal elongation, root growth, and flowering time. More, transgenic Arabidopsis plants expressing a mutant form of ARR4, which was not phosphorylated at the conserved AS residue in the plant's cell-free phosphorylase system, displayed hyposensitive phenotypes to red light in hypocodal elongation and flowering time. This data suggests that the mutant protein exerts a dominant negative effect on the function of endogenous ARR4 and that AS phosphorylation of ARR4 plays an important role in modulating phytochrome B activity. It is interesting that the above-mentioned wild-type and mutant ARR4 overexpressing lines also displayed weak hyposensitive and hypersensitive, respectively, phenotypes in response to cytokinin treatment. Recent studies in protoplasts have shown that the overexpression of type A ARRs represses their own expression, suggesting that type A ARRs may also provide a negative feedback regulation in cytokinin signaling. Because this activity of phytochrome B has been reported and because of the primary cytokinin response properties of ARR4, the photoreceptor may be a target of a cytokinin-modulated two-component signaling system, Figure 4. CRE1-like cytokinin receptors might regulate the activity of ARR4 by his AS phosphorylase and thereby modulate phytochrome B signaling. The phosphorylase may involve the HPT domain protein AHP1, which has the ability to function in a phosphorylate with CRE1 and also acts upstream of ARR4. AHP1 and ARR4 are found in both the cytosol and nucleus. Thus, the AHK-AHP1-ARR4 phosphorylate, as well as the interaction of ARR4 with phytochrome B, may occur in the cytosol to modulate cytosolic, phytochrome B-dependent processes. On the other hand, AHD the 1st of may also be able to transfer the phosphoryl group from a cytosol to the nucleus, where ARR the 4th of may then modulate nuclear, phytochrome B-dependent responses.